Hi, a very good morning to all of you. It's so nice to be in this conference. The topic of my presentation is Fusarium Head Blight Resistance in Wheat bi-parental mapping population. My name is Sharandeep Singh, and I'm a master's candidate under the supervision of Coach Charan Brahad in crop pathology and genetics lab at the University of British Columbia. In the very first slide, I would like to emphasize the importance of wheat crop in Canada and where Canada stands in the international market in wheat industry. Canada stands fifth in terms of production, which is like it produced in 2021 21.7 metric tons of wheat, and is third largest exporter of wheat. In the international market, the Canadian wheat is considered to be the superior quality with 13% protein content, and when you compare it with the average of being 11 to 12%. Canada is, of course, a developed nation, and the noteworthy thing here is that the grain sector is a key driver of Canada's economic growth. There are many production challenges to wheat, talking about the abiotic, drought, and frost damage here. These two are the main. And if you're talking of the biotic stresses, we have insects, mainly the gall midge. Then you have biotic stresses, we have weeds here, and out of the diseases, the viral, fungal, and the bacterial diseases, fungal diseases have proven to be the most detrimental. And out of the 5 priority one, fungal diseases, FHP is the most devastating disease. Um, Now, why FHP is a major concern? First, yes, of course, it diminishes the grain yield, but also it deteriorates the quality of the grains. In the visual appearance, like you see chalky white and shrunken and very like less in weight grains. And if you talk of the other thing, it produces the mycotoxins in the grains. And these mycotoxins, which are the trichothecenes class, produced by this pathogen, it inhibit the protein synthesis, which is like pose serious health risk to animals as well as humans. Until 1990, this disease wasn't that prevalent, and when Canada shifted from conventional tillage to conservational tillage and minimum tillage, this disease being soil-borne, it spread, suddenly took a spike, and until now it has caused losses of more than $1 billion to Canada's economy, with an average of 50 to $150 million every year. And why I call this a major concern, because it costs thousands of dollars loss every farmer every year. Now, when you try to manage it, one of the greatest drawback when you do selections is the strong genotype into environment interaction uh, in this case. Because if you go for selection, even a susceptible line could look resistant, or a resistant line could look susceptible, and vice versa. Talking about the disease cycle, the causal organism, there are many Fusarium species, but Fusarium graminearum is the most prevalent one in Canada. It's soil and residue borne. So during it remains dormant in soil or the residue of the other host plant like corn, rye, barley. During the season when you have healthy wheat crop in your field, it will reinfect that during the flowering stage, and the mode of transmittance is through spores via wind or rain splashes. The spores produced are conidia, which are the asexual spores, and ascospores, which are the sexual spores. I try to make this disease triangle to basically look at like what conditions are necessary for this disease to develop. And if you see Fusarium graminearum virulent pathogen, your hosts are wheat, barley, rye, triticale corn, the grasses, and temperature ranging between 20 to 30 degrees Celsius and high moisture. Symptoms and signs. So the very first symptom in the first picture is you see bleached, chalky white spikelets there. Others are green, and you see three of them are like chalky white. So even in field, if you want to have a look and if you want to recognize FHP, the first thing will be you look at the spike. If you find this bleached spike anywhere, so it's most probably the FHP you're looking at. And then you have, if you second picture, you look, it's the resultant grains, and then The grains also look like light in weight. They are shrunken, chalky white. And third symptom is the mycotoxins, the trichothecenes. When you talk of the Fusarium graminearum, deoxynivalenol is the most common mycotoxin which is produced by the fungus, and it's the most aggressive one. And here's the chemical structure of the dawn. Then, when you talk of the dawn, actually, so when I started studying this disease, for me, I was like, okay, we need to control the disease severity because the spread of the pathogen. But then I came out like, okay, dawn is something has a major role to do in this whole thing because dawn is ahead of the mycelium. When you see the infection, your mycelia follows the dawn. So dawn basically is the first thing that invades the cell, disrupt the cell membrane, kill the cell, and then it is followed by the mycelia of the fungus. So it's an aggressiveness factor. More the dawn, more aggressive is the pathogen, and more will be the spread of the disease. Now, this fungus has two types of mode of nutrition. The first is the biotrophic, like I explained, when dawn invades cell and kills the cell. It's the biotrophic because it's feeding on the living organism. But when some such thing happens, of course, plant also has something to defend, right? And so when the defense system is activated and it leads to like programmed cell death. This programmed cell death again facilitate it's another mode of nutrition of the fungus, which is the necrotrophic phase. So it's again a problem. So talking about its management, first thing come to mind is the fungicide, of course. Yes, we can control it, but there are a lot of expenses. I mean, a single application of a fungicide won't do much to control this disease because the reason is there is always uneven flowering, uneven heading into the field. You have to have multiple applications. And of course, using fungicide has a really bad impact on environment that we all know about it. And then crop rotation, using just crop rotation as a method or strategy of control is not that efficacious. Host resistance is the most efficacious, and I would say environment friendly. You have a certain cultivar which is resistant. So your dependency on fungicides is minimized, and that's a plus point because you are saving a lot of nature around you. Of host resistance in wheat against FHP is basically classified into five main types: type 1, 2, 3, 4, and 5 simply. But type 1 is basically the resistance to the initial contact your pathogen makes with your host, which is wheat in this case, and it's evaluated by spray inoculation. Type 2 is the resistance to spread of the pathogen. Now, when it makes contact with your spike, its resistance to spread within the spike is the type 2, and we evaluate it by point inoculation. Type 3 is the resistance to mycotoxin accumulation. It can either be conversion of the mycotoxin to less toxic form or basically just blocking its accumulation. Type 4 is resistance to kernel infection, Fusarium damaged kernel, or the damage to the grains and its appearance. Type 5 is the disease tolerance. It's basically the inbuilt tolerance that any cultivar or any wheat line that have which can sustain the yield and quality even if you have the high disease pressure and conducive environments. So resistance in wheat against FHP is quantitative, that is small additive genes have cumulative effect against the disease. So here is the mapping population. Paragon is the recipient parent and Watkin 580 is the donor parent. Watkin 580 is a landrace and it was screened earlier for its resistance against FHP and found to be have a good level of resistance against FHP type 2, 3, and 4. So the real size is 94 and we advanced it to F7 where it attained a fairly homozygous state with single seed descent method. And this population was tested in three different environments. One is the greenhouse at UBC, one is the field nursery at Carmen, and other the field nursery at Morden. The methodology used at greenhouse was point inoculation method. So basically, I used the Hamilton syringe, which had Fusarium inoculum inside it, and 10 microconidial suspension was inoculated into the spikelet, like leaving one third of spike from the upper end. And sample size was 94 in two replications, and there was single treatment Fusarium graminearum isolate 109B, which was a single treatment given to all the lines. Now, I recorded the disease severity at 14th, 21st, and 28th day post inoculation. So there is difference between the methodology used in field nurseries and greenhouse. In field nurseries, at Carbon especially, they used corn spawn method, which is more a mimic of natural mode of infection. So you have an infected corn seed with Fusarium, and you spread on the boundaries of the field, which has the mapping population. And the spray inoculation method at modern, which is more of like you spraying the fungicides over the head. Uh, So in the field nurseries, not just disease traits like disease incidence, severity, and efficacy, but also the data on morphological traits like plant height, days to enthesis, and days to heading were also evaluated to make correlations to have better understanding of the phenotype of the disease. So disease severity, how I measured in greenhouse, I just wanted to like simplify it and like that you can understand how we do it in FHP is basically after inoculation, like this is at the 14th day, and you see you have a spike, and you count the total number of spikes. So you divide the disease spikelets by the total number of spikelets, and you take the percentage, and that's your disease severity. And in first case, if you see it's a resistant reaction to FHP, in the second it's a moderately resistant reaction, and in third it's the susceptible reaction to FHP. These are the results from greenhouse, and I tried to sum up my disease severity into this graph, which is area under the disease progress curve, and it is used to combine like multiple observation of disease progress into a single value. It's the measurement of the disease progress. Here, like I measured disease severity over time and space. So in quantitative trait like a resistance to FHB, you have to make observation at multiple days, and then you like to have a better understanding of the plant pathogen interactions, like how they're interacting, how the disease actually progressing. So to better understand this, if you again remember the slide where I showed you the disease severity in a spike, when you have two spikelets infected, then 4, and then 5, and then 19 out of 19. So that's how it progresses over time. And that's the graph. Here, the resistant check is Elson. It means the Elson, in case of Elson, the disease progress is up to this and doesn't go beyond this. And BW8, which is moderately resistant, the disease progress is up to this point. And for the paragon, it's up to this point. But if you look at the lines, the disease progression is basically is following kind of a stable value, and then it shifts abruptly at the end. Also, I have this thing in mind when I was measuring this disease, because when you measure it on the 28th day, your lines or your wheat plant start entering into the senescence, which your plant is turning into yellow because it's entering the maturity stage. And get confused between which is like disease spike. Then you have to open every like okay if the seed is damaged or not. But yeah, that's how we measure the disease progress over time and space. And you have histograms I created for the field trial, which was at Carmen for the same population. And here the resistant check is AAC tenacious, moderately resistant check is FHP 37, and paragon is susceptible paragon. You can see there are lines which are more resistant than the resistant check used, and the lines which are more susceptible. So these are the transgressive segregants, and there is variation within the population for resistance. and the distribution of the population is, in this case of incidence, seems to be a bit of normal. And if you talk of severity, again the checks are same, controls are same, and again there's presence of transgressive segregants and for more resistant and more susceptible. But here the distribution is vaguely bimodal because you have two phases where more than 50% of your lines are into this category and like 30 to 40% in this category. So there is variation within the population and there's segregation of resistant factors, which is good. And disease index is basically product of both incidence and severity. It's a combined picture of both the traits. And then you have field trials of modern incidence. There are two checks. Basically, again, there's presence of transgressive segregants and variation, and for the severity and for the index. Now, the overall idea of the two locations and the results from the two locations is kind of same, that there is variation and there is segregation of the resistant factors and there's presence of transgressive segregants. But there is difference between the distribution. In one you see there's a bimodal, in one you see it's a normal distribution. So basically, it aligns with the fact that in this case, there's strong genotype into environment uh, interaction. Though the genotype is same population, but the environments are different. So we can expect different results from two different environments. These are the results from 2021. So here is the correlation analysis from the carbon and the disease traits that were measured were incidence, severity, and index, and the morphological traits are days to heading and days to enthesis. Days to heading is basically the number of days between your booting and heading stage, and the number of days between heading and enthesis stage is basically days to, like how long. It's about the early and late maturing variety, like how long a certain cultivar or a line takes to reach this stage. The more time one cultivar, one line take to reach the heading or enthesis stage, there's chances that it can escape the disease infection, because at that time, because the time when there is high disease pressure in the conducive environment, cultivar has not reached the stage where it can be infected. So it has a negative and significant correlation, which means more the days to heading, more the days to enthesis, the less will be incidence, less will be severity, less will be index. If you talk of the modern, the trait, morphological trait was plant height. So as your pathogen is soil borne, more the distance between the pathogen and the your head of the plant, your spike, the less will be disease because your pathogen is soil and it has to reach the spike. So incidence is basically the contact, right, that pathogen makes. So incidence and plant height are like negatively and significant correlated. But if you talk of severity, it's not significantly correlated with plant height, which makes sense because the moment your plant your pathogen makes contact with your plant, then the spread of that pathogen within the spike is independent of plant height. It's just a matter of does your pathogen make contact or not. If it does, if it has already made contact, then it doesn't matter what height your head or your spike is. The severity will be independent of the plant height. And then I did the measurement of FTKs and incidence, and FTK and severity has a positive and significant correlation. More the incidence, more the severity, more will be Fusarium damaged kernels. So this is what I have done until yet, and I have three other mapping compilation with Watkin 119058 as a donor parent, and that's my whole project that I will be doing in the coming years. Yeah, I will basically have genotypic and multi-environment phenotypic data for these, and I will look for these SNP markers, which are in close proximity with the resistant regions in the population. And then if we, so if you're able to map QTLs, then we will use this CAPS marker to basically progress these regions or these QTLs into the elite cultivars. Yeah, that's it. Thank you so much. Hope that made sense to you all, and good luck.